Hi, today we are going to look at some worked examples on transcription. I hope you have listened to the transcription lecture. Otherwise, you may follow the link given in the description below and listen to the lecture on transcription before looking at these worked examples. Now, before we go on to the worked examples, let's look at some background. You have a double-stranded DNA. So this is a double-stranded DNA, one strand here, another strand here. Okay, double-stranded DNA, and this DNA can undergo transcription. Transcription is whereby the DNA is used as a template to make an RNA, right? So this is transcription. Following transcription, the RNA is then translated through a process called translation, make polypeptides. So once you have the polypeptides, okay, so these polypeptides are amino acids, and this will then be folded, will undergo folding, okay, to become proteins, mature proteins. So today we are interested in transcription. Okay, we're going to look at some worked examples of transcription. Here we see a double-stranded DNA. So this is the 5' end of the DNA, and this is the 3' end of the DNA. This is one strand. Another strand runs from 3' to 5'. They are anti-parallel, double-stranded. This DNA molecule undergoes transcription. So during transcription, the 3' to 5' DNA template, the 3' to 5' strand is used as a DNA template to make the RNA molecule. So the 3' to 5' DNA strand is known as the template strand because it is used as a template to make RNA. It is also known as the non-coding strand. Whereas the 5' to 3' DNA strand is known as the coding strand. So RNA grows in the 5' to 3' direction. So as you can see here, RNA starts with the 5' end. So when you have a T, here on the DNA strand, so it codes a A onto the RNA strand. A G gives you a C. When you have a T again, you have an A. When you have an A on the DNA strand, you get a U on the RNA strand. Why? Because RNA does not use thymine base, it uses a uracil base. So this is the base pairing. Adenine, okay, from DNA binds to uracil in RNA. Cytosine in DNA binds to guanine. So now let's look at the examples. So here, write down the complementary sequence for the DNA molecules given below. So let's look at the first one. So this is a 5' to 3' DNA strand. So what you are required to give as an answer is the other complementary strand, the 3' to 5' strand. Here you see the 3' to 5' complementary strand. So A binds to T and G binds to C. So this is the base pairing rule. A in DNA will bind to thymine in the other strand of DNA. Guanine will bind to cytosine. So what are these? These lines are the hydrogen bonds. So cytosine and guanine, they have 3 hydrogen bonds between them. Adenine and thymine have 2 hydrogen bonds between them. So this is the complementary sequence. And let's look at the other example, B. So again here, you're given the 5' to 3' sequence. You are required to write the 3' to 5' sequence. So here you are. So G will always bind with C, T will always bind with A, based on the base pairing rules. Let's look at another question. Here you are required to write down the sequence of RNA transcript for the given DNA strands below. So you are given two DNA strands. A is from 3' to 5' direction. You have another DNA strand in B that runs from 5' to 3' direction. As you can see, these are not complementary sequences. They are different DNA strands. You are only shown one strand of the DNA. So let's look at the question A first. Okay. Sequence in A runs from 3' to 5' direction. If you recall, this is the template strand of the DNA, or known as the non-coding strand of the DNA. So the template strand can be used directly to make a RNA transcript. Here we are. RNA grows from 5' to 3' in this direction. So when you have a thymine in DNA, it will code for a adenine in RNA. When you have a adenine in our DNA, it will code for a uracil in RNA. Why is it so? RNA does not use thymine as a base. Instead, it uses a uracil as a base. So adenine from DNA will then bind to uracil in RNA. If you have a cytosine, it will code for a guanine, and when you have a guanine base in DNA, it codes for a cytosine base in RNA. Let's look at B. Now B is interesting. Here you are given the 5' to 3' DNA strand. Now if you recall, this is the coding strand. This is not the template strand. This is the coding strand. So first you need to find the complementary sequence for this DNA strand. Here we are. So DNA is double stranded, so we need to find the complementary sequence that runs from 3' to 5'. So cytosine will bind with a guanine and adenine will bind with a thymine. So this is still DNA strand. This is the template. This is the template strand. The template strand can now be used to make a RNA transcript. Here we are. So the RNA transcript, when you have a guanine, it codes for a cytosine. We know that. When you have adenine, it codes for a uracil. When you have a thymine, it codes for a adenine. When you have a cytosine, it codes for a guanine. So now let me tell you a shortcut. Do you see the similarity between this strand and this strand? Can you tell me a similarity between these two strands? Can you see that they look the same, except that all the thymine are converted to uracil? See that whenever you have a thymine, it gets converted to uracil. Other than that, the sequences are the same. So this is a shortcut that you can use if you are given a DNA strand that starts from a 5' and ends with a 3'. if you are required to write down the RNA transcript, you can copy the same sequence and convert all the thymine to a uracil. So that is a shortcut method. In this question, you are given a RNA molecule, an mRNA molecule. Now, this is interesting. You are required to write down the DNA sequence, the non-coding strand sequence, which coded for this mRNA. Now, if you recall, you have the coding sequence, DNA that runs from 5' to 3', and then you have the non-coding sequence that runs from 3' to 5'. This is the template strand. And from here, you can make your RNA, right? So now you are given your RNA and you are asked to write down this sequence, the template sequence. So how do you do that? Okay, so here we are. We have the mRNA sequence here. Okay, now we need to find out what is the template sequence. So here, so this is 5', so this is going to be 3' because your template strand, also known as the non-coding strand, runs from 3' to 5'. So again, this is just base pairing, complementary base pairing. An adenine will bind to a thymine, a uracil will bind to a adenine, a guanine will bind to a cytosine, and a cytosine will bind to a guanine. As you can see, the DNA template strand is complementary to the mRNA strand because the template strand is used as a template to make your RNA transcript. Thank you very much. I hope you have understood how to write complementary DNA sequences, how to derive the RNA transcript sequence from a given DNA strand, and also how to work backwards or reverse engineer to find out the DNA sequences from a given RNA sequence. Thank you.